Get ready to enter a brave new world with your host, Vasant Dhar.
For more color on the podcast and additional commentary, please subscribe to my newsletter at vasantdhar.substack.com. Hello, and welcome to Brave New World. My guest today is Leslie Kay, Professor of Psychology and Neuroscience at the University of Chicago. Leslie studies olfaction, currently one of the most exciting areas of research in perception. So that's what we're going to talk about today, smell, which is also an area of inquiry in which I am deeply engaged at the moment. Leslie, welcome to Brave New World. Delighted to have you on the show.
Thank you. I'm really excited to be here. I'm a big fan.
Thank you. So, before we get into the substance of the day, I usually ask people to introduce themselves and just tell us a little bit more about, what kind of influenced you growing up and how you ended up doing what you do now.
You might say I'm kind of an accidental scientist, I grew up in Los Angeles, in the San Fernando Valley. This was before there were Valley Girls, in fact. I went to LA City schools. I had what was then a normal, good education. I think it's also important to say that all of my grandparents were immigrants who met in this country. All four of their families fled pogroms in Eastern Europe and came to the United States for a better life. And no one in my family was a scientist. We have people who built small businesses, teachers, lawyers, politicians, pretty much everything but scientists. And so I kind of came to it accidentally. In the seventh grade, I was really good at math, and the guidance counselor called me in to talk to me about whether I wanted to enroll in the advanced math class instead of the regular math class. It sounded good to me, but he said to me, well, a lot of boys want to take this class. And so if you really want to take it, we'll put you in it if you really think you're going to be a scientist or a mathematician when you grow up. I was 12. I had no idea. I wanted to be a writer. But I loved math, so for the first time in my memory, I stayed up all night worrying about whether I could lie to this man. And for some reason, I never asked my parents. I'm not sure why. So I went in the next day and I lied. And I said, yes, I want to be a scientist. So I took this class and I got tons of great math education. I really loved math and science in high school. I ended up doing my undergraduate education at a liberal arts college, St. John's College, which is famous for making you study everything, math, science, philosophy, literature, music. It was perfect for me. And that was in Santa Fe, New Mexico. So I kind of accidentally got a job working at Los Alamos Labs when I graduated, and for the summer before my senior year, I worked for the original, 6 people in a trailer for the GenBank project, which became the United States Genetic Sequences Database. And there I met a lot of scientists who had studied biophysics, who had gone, who'd done their degrees at Berkeley and were working on really interesting dynamical systems type things. But I was mainly reading genetics papers and annotating gene sequences.
And this is like you were in your early 20s?
I was, my early 20s. So there were really 6 of us in a trailer on the edge of a what's it called? A giant arroyo. I forget the name for the bigger thing that's not an arroyo. And it was beautiful and it was fun and it was, the early 80s. And Walter Goad, who started GenBank, thought, wow, all these people are publishing DNA sequences. We should store them somewhere. So that was it. Now it's run by the National Library of Medicine. And I started grad school at Berkeley, but I had in biophysics, but I had no idea what I wanted to do. I had no career counseling. So I took some time off and worked as a database programmer for an insurance company in the late 80s. And I ended up going back after about 5 years to finish my PhD. Well, really to start it. And my PhD advisor, another Walter Freeman, really had a profound influence on how I think about brains. I came to him and I said, I'm more of a mathematician. I take a ton of math classes. I really want to do modeling of brains because he was doing a lot of very complex modeling that still is more complex than anyone else does. And he said, we'll see. I mean, in my memory, he's chewing on a cigar in the corner of his mouth because he always had one. I don't know if he really did. And he handed me some data, which I started to analyze, and I found something interesting in the data. And then I had to do an experiment and I became a physiologist. So really, this was a completely accidental series of events that led me to that place.
I was chuckling to myself when you were talking about, chewing on a cigar and saying, we'll see, because I had like a very similar experience in my first aha moment that got me into AI was two people chewing on cigars saying something similar.
Yeah. Well, we'll see what you can do with this young man, right?
Yeah, exactly.
Yeah.
No, what I was going to ask you was, so what was Walter doing, in terms of understanding the brain and especially smell, which is, the topic of the day? And how did you get into studying olfaction?
Yeah. So, I liked him and I liked his lab because it seemed exciting. He also, like myself, loved doing other things. His bookshelves in his office were full of philosophy and poetry books. And I don't know, I tell people I picked him for all the wrong reasons, but he was perfect. And so I learned how, from the very beginning when I started learning about brains, I learned how to think of them as this, distributed dynamical system that was capable of very complex activity. And he was really one of the first people in neuroscience saying the brain is, a complex dynamical system and has structure like it should work in chaotic regimes. And he went hard on the chaos model. And that was not very attractive to neuroscientists in the United States in the 1980s.
So say that in English, right? Because you said a lot, right? You said neurodynamical systems and you said chaos. So reduce that to English.
Yeah. So dynamical systems is, we can think of, a system as kind of an assembly line, right? Where processes get accomplished in parts and sequence. But a dynamical system is something that accomplishes the task at hand by having a lot of, let's do it mechanically, moving parts interacting with each other to produce the outcome, right? More like, how you might throw a baseball. It's not, if you're good at it, which I'm not, you know a lot of things and you can write down the steps to do it, but it doesn't work at all if you just follow the steps. It's more about training all the different muscles and your reaction times, and they all kind of work together for the good pitch to emerge. So this is, how I think about brains, how I learned from Walter to think about brains. And at the time... So
let me just interrupt just to clarify just for myself. So the way I hear you describe this dynamical system is that I think what you're saying is that we perceive the world around us sort of as active participants, who are taking in information and maybe making some sense of it and then taking another stab at it and making some more sense of it. Ultimately, sort of somehow the brain says, oh yeah, that's a rabbit, or that smells like Provence, or whatever, right? Is that kind of what you're saying? That it's not just a passive receptor of information that's then interpreted.
Yeah. And, when we get more into how the olfactory system is structured, it's not structured in a way where it follows smoothly an idea like an assembly line. Like, here's the input, then this is processed and it's passed on to the next stage and that kind of thing. There's nothing in the anatomy of the brain that indicates it should work like that. but as neuroscientists, we kind of are taught, and it's the fashion, at least in U.S. neuroscience, to go hard on the information model, which is this passing of information from one stage to the next. And when Walter started his work at the time, neuroscientists Linda Buck and Richard Axel had just discovered the olfactory receptor genes, right? It was in 1991. I think that was right when I joined the lab.
And when did they get the Nobel Prize for that? Was that 10, 12, 15 years later?
Yeah, something like that. Yeah. And it absolutely was a game changer for understanding the sense of smell. So imagine if, for 60 years or 100 years, people had been studying vision without knowing that we have 3 color receptors, right? What stories would we have made about how vision works without that knowledge? So the knowledge, of how many olfactory receptors there are, and there are hundreds in humans, and 400 as opposed to 3 color receptors. So it's this huge multidimensional input space that then is somehow, accepted by our brains in among the ongoing activity and turned into a perception, And so that's where I sit, is kind of at the space between the input and the perception.
Leslie, I've been studying smell over the last few years with my data science PhD student, Vivek Agarwal, and my neuroscientist colleague, Dima Rinberg, who runs the Rinberg lab at the NYU Medical Center. And I'm fascinated by how smell works. So what do we know about how we smell?
Yeah, it's, I think we benefited by not knowing about the receptors for such a long time, because there were a lot of theories, a lot of competing theories about how smell works. The problem with the discovery of the receptors is that people dove into, okay, now we know, right? In fact, the Nobel Prize announcement about the discovery of the receptor gene family said basically that smell was solved. And, we all know that vision is not solved by knowing what kind of color receptors we have. But so what do we know? I mean, we know there's this very high dimensional but ordered input to the system. So the nose, you breathe in, you get lots of molecules. Those molecules attach to or they activate receptors. Sometimes they inhibit them. And each type of receptor kind of sends its own parceled part of the signal to a spot or a couple of spots in the olfactory bulb. And, these receptors are not sensitive to whole molecules. They're sensitive to parts of molecules. And so, but most of our natural odors, right, are mixtures of tens or hundreds of different types of molecules, so which have different volatilities and different sorption properties and different electrochemical properties. So it's a really complex problem added on top of these many receptors. In a rat or a mouse, it's over 1,000 types of receptors. For humans, it's close to 400. But once you get inside the olfactory bulb, the receptor layer is kind of washed away, like in the very first stage of processing. So this beautiful order is sort of lost in how the cells respond. So if an animal's anesthetized and you record a neuron that gets its input from a single spot in the olfactory bulb called the glomerulus, it responds really nicely to that odor, or if the odor doesn't mean anything to the animal. But once you train the animal that the odor means something, and I would challenge you to find an odor that doesn't feel like it means something, right? We'll talk about that in a minute. The response of that cell to that odor changes. So the cells that are getting input directly from the nose tell you more about what the odor means and what the animal is doing than it does about the chemical composition of the odor, which is really crazy. When I tell this to undergraduates in my classes, I pause, and at first they're a little puzzled, and then they go, wait a minute,
Yeah, so I was going to ask you, like, what does that mean? Like, what is it? What is the meaning of an odor?
The meaning of that is we don't have objective sensory information in our brains when we're awake. If we're anesthetized, you can get really nice responses. It can look like we do if we arrange the behavioral circumstances correctly. But after you get through the input layer, you can see like odor-dependent patterns of activation of the olfactory bulb. And this is a lot of what Demo works on, really beautiful, analysis of how the glomerular sheet, the glomeruli are the little spots where the receptors provide their input. But after you get past that layer, it's like that layer never existed. So the question is, is the layer just an accident of wiring? And it wouldn't really matter. That's more of a philosophical question.
When you say that layer never existed, I guess that's not entirely true, because even in your own work, what you say is that you get that initial sniff and, the brain does something with it, and maybe the brain says, well, I need more information. And so you get more sniffing as opposed to the brain saying, all right, I'm done. I've recognized it, right? So
Right. But what does it mean about recognizing it, right? My brain is certainly not saying, oh, that's, 1 octanol. My brain is saying, oh, I know that smell. What was that? When did I smell that? Oh, yeah, now I remember when I smelled that. Or I remember a word that goes with that smell. We don't identify
or not, right? I mean,
or not. We might say
it's maybe something just entirely new, and and that's what I mean by, like, what does meaning really mean in smell, right? That you smell something new, and the brain kind of says, okay, I smell it, but I have no idea what it means. I've never smelt it before, but here it is, right?
Right, and that's going to involve the collaboration between not just the olfactory bulbs, but also the olfactory bulbs, the rest of the olfactory system, the limbic system to which these cells send output. So the very same cells that have their receiving end right at the surface of the olfactory bulb receiving the sensory nerve input have their output ends to every part almost of the olfactory system and to the limbic system, to the amygdala, to the entorhinal cortex, which goes into the hippocampal formation. So all these areas that are essential to thought and to our production of meaning, right? Our emotions and our thoughts and our memories associated with odors, those are part of the olfactory system. This whole system used to be called the rhinencephalon, actually, the smell brain. And
you mean the limbic system?
The limbic system plus the olfactory bulb. And depending on which book you pick up to read, to look up what the limbic system is, some of them include also the olfactory bulbs and the piriform cortex, which is it's called olfactory cortex, but it's not really.
Now, just to make this totally accessible to people who don't know anything about smell, I think what you're saying is that smell is the only sense that connects directly to the limbic system, right? That all other senses go to the thalamus, through the thalamus, which is sort of just, my simplistic interpretation is there's this relay station that takes in stuff and figures out what to pass on to, the neurocortex and what, so it does some mediation, whereas smell goes directly after just 1 or 2 synapses directly to the limbic system. Have I described it more or less correctly?
That's correct, except I think you maybe described the thalamus a little too simplistically. So Murray Sherman, who's also at the University of Chicago, would have a bone to pick with that explanation. But it's a perfectly fair one, because that's what everything says that we read, that the thalamus is basically a place where sensory information stops to get a cup of coffee, and then it goes on to do the real work in cortex. There's more input to the thalamus from the brain than there is from the sensory inputs and to all of the sensory areas. And there's all kinds of very cool modulatory stuff that's happening. So it's not just, it comes here, does a little bit of processing, and then it goes on to do real processing. It's this, again, dynamical interplay, and it depends on the state of the individual, right? Whether they're asleep, awake, under anesthesia, paying attention, not paying attention. The whole character of the visual thalamus, for instance, the lateral geniculate nucleus the whole character of how the cells respond to input changes depending on the state of the animal. And the cool thing is the olfactory bulb is exactly like that. About, almost 20 years ago now, Murray and I wrote a paper. I forget the title actually, but it was, is the olfactory bulb essentially a thalamus? And if you look at all the synapses and the connections and the whole structure, it is exactly a thalamus. So yes, the olfactory nerve goes directly to the olfactory bulb, what we might call the olfactory thalamus, just like the visual nerve goes directly to the thalamus, the lateral geniculate nucleus. But from there, just a ton of stuff happens to it. So when it leaves the olfactory bulb, I'm talking about it like it does this in sequence. It's doing it all at the same time. Yeah. When it leaves the olfactory bulb, it's a completely different signal than it was when it got there. And the olfactory bulb gets, tons of input from all over the limbic system and cortical areas and every neuromodulator system. So it's this huge hand of God coming in and adjusting how the olfactory bulb is going to receive this sensory input.
So Leslie, how do you actually observe all this stuff, right? At Dima's lab, we've got mice and we've kind of, got cameras pointed at the glomerulus and we're observing neural activity. And, so that's the method of choice primarily for us. What have your methods looked like where you've been able to observe this sort of neural activity in response to odorants?
Yeah, mine is pretty old school. We basically stick a wire in a brain and attach an amplifier to it. There's more electronics involved than that, but the signals we're looking at can be picked up by, electroconductive wires. Mainly we use stainless steel. We've used some other metals. So they pick up voltage changes. And the reason that voltage changes are important is that when neurons are active, the way they're active is they're moving positive and negative ions in and out across the cell membrane. And that makes the inside more positive or the inside more negative relative to the sort of, salt sea that these cells sit in all the time. And the difference in voltage across the membrane makes a signal. So it makes each cell makes a teeny tiny little field around itself. And the cool thing is that cortex has geometry. It has bunches of cells that are all lined up. So some of them, depending on their architecture, are lined up. They're sort of parallel. And so they make these beautiful fields that are all perfectly aligned, or more or less perfectly. So you stick a wire down there and all those fields sum up and create, sort of a summed signal. So we put a wire in there, say a giant wire, like 100 microns in diameter. So that's about 1/10 of a millimeter. It's exactly 1/10 of a millimeter. We put it in there and we can record the collective activity from, hundreds to thousands of neurons, depending on kind of where we're positioned. And so what I mean by collective activity, it's the activity that's the same or shared among that group. All these neurons are connected together, right? So the cells that receive the sensory input from the nose in the olfactory bulb, they're called mitral cells because they look like a mitre, an upside down triangle. And they connect not only with their targets, but they have these sort of long lateral dendrites that they have a dendrite at the top that gets the sensory input, and they have these lateral dendrites that connect to hundreds of tiny neurons called granule cells. And the mitral cells excite the granule cells. The granule cells inhibit the mitral cells. And when you wire them up together, what you get is this sort of sequence of mitral cell excites granule cell, which makes the granule cell inhibit the mitral cell, which makes the mitral cell stop exciting the granule cell, which makes the granule cell stop inhibiting the mitral cell, and then it all starts over. Because the mitral cell is always getting this, sensory drive, right? It's always got some energy coming into it. So it ends up, it's what's called a classical negative feedback circuit, that you get excitation, inhibition, disexcitation, disinhibition, and then you're back to the start. So you get an oscillation, like a sine wave.
I see. And so if a subject is smelling different kinds of things, you're going to get different kind of behavior generated at the system level. You
do, but not like the frequency doesn't change in the oscillation. What happens is if we look at the interaction between all the parts of the brain, and this is where machine learning tools, are starting to come in handy, because it's subtle, right? Because these signals are recording thousands of neurons. And so they're saying, what is the olfactory bulb thinking right now? And this coherent signal is functional. It's not about the signal itself. It's not about the oscillation. Those are helpful for us. It's about what the oscillation indicates to us about what the neurons are doing. So when the oscillation is, bigger, that means more neurons are firing in the same time window. And so when neurons fire in the same time window at their other ends down to the next cortical area, spikes are more likely to arrive at the same time, because every neuron's axon is a little different diameter. They have different amounts of insulation. The signal travels different speed in each one. So if you're trying to drive a neuron in the next area of cortex, if you're trying to excite it enough, it needs input in a small time window, a few milliseconds, from a bunch of cells for it to respond.
So what does more activity mean? Does it mean that there's more uncertainty, that the brain's doing more work and trying to decipher the signal? Like, how are you interpreting this data?
So this goes back to my postdoctoral research and, the accidents of science, who you work next to, right? So I did my postdoc work in Gilles Laurent's lab at Caltech, and I was there as a Sloan Fellow in the brand new computational neuroscience program. And, John Hopfield, Christof Koch, a whole bunch of people and a bunch of my contemporaries who ended up, going and sort of staffing computational neuroscience across the world. We were all talking to each other. And one of my lab mates, the lab I was in did insect olfaction. And I was the only person working on rats. I was recording neurons in the olfactory bulb to find these objective responses, which I never found. I only found that the neurons changed their response when I changed the meaning of the odor. My next door neighbor, basically in the lab, Mark Stopfer, was working on honeybees and showed that if you disable these oscillations in the honeybee antennal lobe, which is really almost the same as an olfactory bulb, the honeybees can no longer tell the difference between similar odors. They can still tell the difference between odors that are very different, but these oscillations appeared to be necessary for the honeybees to tell the difference between similar ones. So this got us thinking about, are the oscillations about the coordination of neurons? So when they're bigger, it doesn't mean that the neurons are firing more. It just means they're firing more precisely in smaller time windows.
So they're trying to discriminate finally.
Yeah.
Finally.
And so, at the same time, I was collaborating with people at UCLA who had this Zoli Nusser, who's now at the in Hungary, in Budapest, knockout mice that they were studying something else about the mice, but they discovered they also had enormous what these oscillations are called gamma oscillations. They had pathologically large gamma oscillations in the olfactory bulb. So I tested these mice to see what their discrimination abilities were, and they were better than control mice at discriminating odors.
Talking about discriminating odors, you obviously have heard of Joy Milne, right? The Scotswoman who can identify Parkinson's.
Yes.
Right. So, I mean, when I first came across that a few years ago, that just completely blew my mind, right? Because they did a controlled experiment. So the experiment was that I think a dozen or 2 dozen people with Alzheimer's and without wore a T-shirt for some period of time. It was cut down the middle and they sent it to Joy. And she had a perfect record except for one false positive, and that person developed Alzheimer's or was diagnosed with Alzheimer's.
Parkinson's.
So,
right?
sorry, Parkinson's.
Yeah.
The following year, right? So this is like a perfect track record, right? In a human, and this is, and the story is that she had smelled this on her husband 12 years before he was diagnosed. And she only realized she had this ability when she went to a support group of people with Parkinson's, and she said she smelled it on everyone. Right. And that's what then led to this subsequent experiment. So to me, that's, really existence proof that biological organisms, I mean, humans, and therefore potentially other animals, have this ability to sense something perhaps years before any kind of biomarker is identified with that. So how do you think about this? Like, where do we stand with respect to the role of smell in health?
Oh, yeah. We're in really bad shape.
You mean we're in very early stages?
Yes. And we should be further along. The problem is that, people have this illusion that smell isn't important. I mean, really, you can get along in modern life if you lose your sense of smell. That's not a problem. And most people actually don't even know they've lost their sense of smell until it gets very, very bad. And some people don't know at all. And that's actually a very cool kind of interaction of sensory systems. So we think, it's not really important. It's vestigial in humans. We enjoy smells, but we spend more money on scents each year than we spend on tequila, right? So it's clearly important to us to some degree. And it's also smell dysfunction it decreases with age normally, and that's not pathological. That's just your sensory receptor stem cells kind of wearing out as that's what is thought to be the cause of age-related smell decrease. So it just things don't smell as strong as you get older. But with neurodegenerative diseases like Parkinson's disease, Alzheimer's, Huntington's, Lewy body dementia, frontotemporal dementia, lots of diseases, most of which end in dementia, appear about a decade earlier with smell loss. And so the problem is we don't really know how to test for it. We have some olfactory tests, but they're more about how good is your smell. It doesn't test the fine points of how we smell. And there are some differences in what you lose with different diseases. So there's a lot of work now based on looking for, trying to develop a test that's specific. And there's one, a recently published paper. It's called the Aromha test, AROMHA. The paper is published last year, I think. The lead author on that is Mark Albers at Mass General. And to be transparent, I'm also collaborating with him on trying to use this smell test. We're trying to get money to recruit people on the south side of Chicago. So it's a test that tests like how well you can smell. So that's your smell thresholds, like how strong does this smell? And then has you do a lot of more cognitive tests, telling the difference between odors, remembering if you smelled an odor, 30 seconds later, more cognitive olfactory tests like that, which are differently affected in different neurodegenerative diseases. So, and this test, they send you a card. It's a big card, comes in a plastic sleeve, and it has little like stickers that you lift up and smell. And you complete everything online. So it can be done completely unsupervised at home. And this paper shows the ability to identify cognitive decline. So that's the first step. Can you identify not just I can't smell very well, but can you identify cognitive decline? And it looks like it can. So more research is needed, of course.
That's very interesting. And it may not just be cognitive decline. It may be other kinds of diseases as well. I mean, it's a question of like, what are you actually looking for? And so far we haven't been looking at all, because especially for these generative diseases that show up so much later, when the horse has left the barn. And so we don't actually gather any data in the early stages because we didn't know we should have been looking.
So I'll make a wager here that you have never, as a part of your routine medical care or physical, had a smell test.
Yeah, never. And that's what I find amazing, right? That there is no smell test, right? We have vision tests, we have hearing tests.
Right. And in fact, scientists and physicians have sort of teamed up to try to get the U.S. Medical Association, writ large, to recognize smell dysfunction as an important part of disease and smell health as an important part of standard medical care, to find a way to include smell tests in regular healthcare. There's in fact no insurance code or Medicare code for administering a smell test. If you go to an ENT and say, I've lost my sense of smell, and they give you this little scratch and sniff smell test called the UPSIT test, it tests whether you can recognize odors. It doesn't test other things like thresholds. And there are a couple of other tests that can test thresholds. So if the doctor gives that to you, they either have to pay for it out of pocket, from the medical center funds, or there are some cases where it can be classified as an expense, but there's no code to charge it back to the insurance company.
So when do you think that will change? Any prediction?
I don't know.
Because clearly, this is something we're not looking for at all, which we should be, and at the very least, we should be gathering data about this sense.
Right. So if you knew, here's the last 5 smell tests I had, and all of a sudden it dropped 2 points, and I haven't had a cold. I don't have, a sinus infection. What's going on? Then you might do more tests and you might catch things really early. But yeah, I mean, we go to, as a professional organization, the American or the Association for Chemoreception Sciences, ACHEMS. One day each year we'd have a Hill Day and we go and talk to our representatives, and they're very receptive. But to be frank, there's a lot more going on right now with medical research. But I think we just keep plugging away. Actually, I have. Along those lines, I have a book under contract with Basic Books and Penguin UK about smell. It should be coming out late 2027, but it's designed to help people understand how smell works.
Excellent. Good luck with that writing project. So the whole area of figuring out one's health based on smell is a big frontier of research, isn't it?
Absolutely. And it's funny because, bomb sniffing dogs, they are really good at it. It is very difficult to make a machine that can be as good as a dog, which gets to how complicated the olfactory system is. It's not really just a chemical detector. It's used in service of detecting chemicals, but the way it does that is to attach some meaning to it, right? The meaning that gets, for the dog, it's I get a treat, or I get to play with my toy if I find the bomb smell. And so it's not I'm looking for 1 octanol. It's I am trying to get a treat. And so when we start thinking of the olfactory system, and I think all systems, as evolved in service of taking the external environment and behaving in it in a way that's advantageous, right? Getting food, finding mates, avoiding predators, living long enough to have children and grandchildren. That's what smell, is evolved in service of. And in fact, it might have evolved for a completely different reason. So Lucia Jacobs, she's now emerita professor from UC Berkeley. She's an evolutionary biologist, studies learning and memory and the hippocampus, and she's moved into smell. And she wrote this beautiful paper in 2012 talking about how the olfactory bulb evolved as a scaffold for spatial cognition. And, if you think about our vertebrate ancestors were fish, right? And what is space to a fish? Space is food, where my food is, a location is where my food is, where my, if you're a schooling fish, where my mates are, and, other landmarks, which, their vision is not terrific. They can smell really well. Fish are great at smelling, and they're really good at smelling food. But that place, if we're standing on the shore, it's not where we are all the time. It moves, right? So where the hippocampus evolved as, sort of, or we look at it as using a lot of, it uses multisensory landmarks, but it uses vision really heavily. At least our experiments for it do. The olfactory bulb was the place where space use evolved. And she compares, a bunch of different animals. And then when they moved onto land, they still continued to use the olfactory bulb for space. But the hippocampus started to get bigger. And if you compare different animals' predation strategies, how they use space to find their food and their mates, actually depending on their space use and the kind of use of that space where it might involve smell cues, the olfactory bulb is bigger in those species relative to the hippocampus. So the olfactory system is, not about odor identification, really, although that's, what we use to test animals. It does a whole bunch of other things. And it may, in fact, sit at the foundation of how we think because of its connection with these areas of the limbic system.
So that segues nicely into where I wanted to go next, which is how does it relate to consciousness? And in preparation for this conversation, I asked ChatGPT. I couldn't resist, obviously.
I should have done that.
So I said, is smell related to consciousness at all? And it said yes. And then it gave me a whole bunch of literature, and you'll be pleased to hear that it actually listed you as one of the people in this space, and it pointed to one of your articles.
Interesting.
So I'll read you like the first part of it because it's a longish response. It says, number one, olfaction is closely tied to brain structures involved in consciousness. Unlike other senses, the olfactory system bypasses the thalamus, connects directly to the limbic system, blah, 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 blah. And these areas not involved not only in emotion and memory, but also implicated in core features of conscious experience like self-awareness, emotional valence, episodic memory, sense of time and continuity. And, and then it goes and lists other things like smell is subliminal. And then it raised a really interesting question, which I hadn't thought about because, I remember listening to a panel discussion among some of my colleagues last year about whether perception is necessary for consciousness. ChatGPT actually says, is consciousness necessary for perception? Was the question it raised, which I thought was fascinating, right? That is, can you actually have perception without consciousness? So how do you think about it? How do you think about this relationship between smell and consciousness?
I think about it a lot, but the reason I laughed when you said it cited one of my articles is that I typically forbid what I call the C word in my lab, because students come in and they want to study, they want to be neuroscientists and study consciousness. And my attitude is you have to learn a lot of basic science first before you can ask that question intelligently. But we actually talk about it all the time. We just don't use that word.
So why don't you use that word, right? Because I've had a couple of conversations about consciousness with, Dave Chalmers, the philosopher, and Anil Seth, and maybe a few other people. And they've got sort of, strong theories about, well, I shouldn't say strong theories, but they've got theories about what they mean by consciousness. But why do you avoid that? Is it because it's just too grand or
Yeah, it's too big, it's too big a word. I mean, it's so big that it means nothing. Because if you're a neuroscientist and you say, I'm studying consciousness, what you're really studying is brain activity. You could call it consciousness, right? And I think and this is where dynamical systems comes in, right? So causal mechanistic processes exist at sort of every level of scale, right? In molecules, in individual neurons, in ion channels, in networks of neurons, in brain areas, in connections of brain areas, in whole brains, and how those brains interact with other brains, et cetera, et cetera, et cetera. And at each of those places, you can assign activity measures to correlate with some measure of, and I'm going to go low here, like let's just say perception. Let's assume that at least in mammals that perception correlates with some level of consciousness. But it doesn't explain how we get to it, right? And because what we have is all of these causal influences operating at different levels, but affecting each other. Like, for example, you could smell something and have a thought, right? Oh, I smell grapefruit. You smell grapefruit, I smell grapefruit. Our brains do different things with it because you probably don't have the pleasant childhood breakfast table memories that I have with my parents and my brothers, right? Eating grapefruit with the little spoon that cuts the grapefruit pieces out for you. But the smell of grapefruit brings that all back for me, right? It's bringing that all back for me is, somehow an evocation of that past memory, which becomes conscious, right? I pay attention to it. That makes my heart beat faster or makes me feel happy, changes my physiological state in a way which feeds back to my brain, understands that that's happening. It says, oh, now I'm happy. And that changes how the system works. But what's happened there is something has emerged into consciousness and then gone back and affected my neural system. We don't know how that happens, except through an explanation that has them happening in parallel, right? So if we go to, Patricia Churchland, we could say, okay, all this brain activity is happening and I'm kind of along for the ride. Right. My sense of it is simply, the patina on top of all of this stuff that's happening. But what is that sense? It's very difficult to assign it to a neural function. So it's, in dynamical systems, we call this an emergent process, right? It emerges from the activity of lower levels, but it also affects the emergence of it affects my acknowledgement of it, and the emergence of it affects how that happens. So now that I've told this grapefruit story, which, I just kind of was thinking about it last night, and I kind of, I picked grapefruit and I said, do I have an association with grapefruit? Oh, yeah. So now grapefruit for me is going to be, really strongly associated when I drink the grapefruit flavored soda water. I'm going to have that experience now where I didn't used to have it. And that's all because I had a realization, I had a conscious, and I'm using it in a really basic way. Although I was trained deeply in philosophy at St. John's College, I don't count myself as a philosopher. And, so I'm using it in a really basic way. I'm conscious of what I thought about and how I felt about it. It's present to me, and that influences how my brain works about it. So that's where I think, we can study consciousness, and smell is such a big part of that, right? It's not really an accident that smell and breathing, especially through the nose, is a big part of many religions. So if you want to go from consciousness into spirituality, which tend to get kind of mixed up and confused together, not for, bad reasons, this entrainment that smell and nasal breathing can produce can entrain olfactory areas, but also entrain them with other areas that are sort of core to our sense of being. The hippocampus, the amygdala, all these cortical areas connected with them. And I had a student who did, Andrew Sheriff, who's now working with Christina Zolano at Northwestern on human olfaction. But he did this beautiful set of experiments where he recorded in olfactory, hippocampal, visual, and there was something else, areas in a rat's brain, and had them just foraging for chocolate sprinkles, either in the dark with a couple of prominent olfactory cues at right angles to each other, or in the light with a couple of visual cues at dim light at right angles to each other. And the animals did the same behavior, but their brains did it completely differently. And the biggest factor in syncing up their brains in different configurations was their breath. We had a nasal breathing signal. We had all these brain electrodes, and the rhythm of breathing synced up pretty much the same, either in the dark or in dim light, just sitting in a cage. But when they're out doing these tasks, their brains were connected very differently, depending on whether the lights were on or off, and whether there were smells there or not. And so, and that all happened at the rhythm of breathing. So it says to me that, the breath that you take in through your nose, now we can breathe through our mouths also. Rats can't breathe through their mouths. But the air that you breathe in through your nose has a profound effect on how different areas of the brain are connected.
That is absolutely fascinating, this connection between breath and brain activity. And of course, breath, what we call, quote unquote, the sniff cycle, plays a key role in how we collect smell data from mice by observing their neural responses as they sniff various things.
So one of the things we tend to think about in olfaction is the sniff, right? So rats sniff, and most neuroscientists, not all of them, most olfactory neuroscientists treat the sniff as kind of a unitary thing. A sniff is a sniff. It's different than breathing. A sniff draws the air up to the olfactory receptors, where just breathing, it only kind of grazes them. That's why you can smell smoke when you're not sniffing. But then if you really want to see where it is, you start to sniff, right? So rats do this too. And Max Mozell had a hypothesis that the nose, in particular the sensory epithelium, is like a gas chromatograph. That it's covered with mucus, which absorbs chemicals into this sort of watery medium. And a gas chromatograph has kind of a sorption column that traps molecules that, dependent on their sorption properties, right? And that separates them because they get absorbed at different times and get released at different times. So he had this theory, and he showed that if you put a frog nasal cavity on a gas chromatograph, it works pretty well. So my student, Daniel Rojas Libano, who's now a professor in Santiago, he decided to test this. So he trained rats. He said, well, rats, if they're picking out something that's highly sorptive, that gets into water easier, they should use fast sniffs. And if they're wanting to pay attention to something that's hard to get into the mucus, they should slow down their breathing. And after they learn to find these odors in mixtures, so we give them one target, either a high or a low sorption odorant, and then they sniff all these different mixtures to find which one it's in. They sniff faster or sniff with faster flow when they're looking for the high sorption molecule than when they're looking for the low sorption one. This is such a beautiful set of experiments, and totally due to him, came to me with this question and said, I want to answer it. And he did. So it showed, and in fact, they change the flow rate across sniffs, right? The first 2 sniffs are different from the later sniffs. And we do this too. If you're smelling something like a glass of wine and, you're a sommelier or pretending to be one, you sniff it and you smell something and somebody says, oh, do you smell the raspberry in there? You might take another sniff, and what you don't know that you're doing, but you're really doing, is you're changing the airflow in your nose to look for something else. And so we called Daniel's experiment the Where's Waldo experiment. If you familiar with Where's Waldo, these children's books, right? Yeah. So if you're not looking for the guy with the red and white striped shirt and the funny hat and stuff, you're never going to find Waldo. But if you're looking for him, you move your eyes and focus your eyes on different features in such a way that you can find him, right? And so we do that with our noses. We just don't know that we do most of the time.
Fascinating. So Leslie, you wrote a paper about the impacts of COVID on smell and, health in general. Say a little bit about that.
Yeah, April of 2020, March of 2020, we're all sitting in our kitchens trying to work. And I got invited to a Slack group called the Global Consortium on Chemosensory Research. And ended up being about 600 scientists who study taste and smell. So I opened up the feed, and really to my instant horror, my colleagues in Europe were reporting that people with COVID were losing their sense of smell. And sometimes it was the only symptom they had. And you know how that expression, the bottom dropped out? It felt like that. Free fall. I was just terrified because of this connection I know about between smell dysfunction and neurological disorder. And, so I spent the next several months still in my kitchen trying to work, but also attending Zoom meetings with the autopsy team at the hospital here and talking to colleagues across the world. I couldn't sleep for months. So this was because of this connection. And the thing is, if we want to study that, if we want to study whether an insult to the nose could affect the brain, we'd get a lot of people sick with the virus at the very same time, right? So, suddenly I realized we were in the middle of a giant experiment, because this has been a theory of why all these neurodegenerative diseases start with the loss of smell. That at least with some of them, there may be some viral insult that hits the olfactory system and starts a slow degenerative cascade, basically, that works its way to the limbic system. We know this happens in Parkinson's disease. That's the march starts in the olfactory bulb for many cases. And animal models show if you seed, damage in the olfactory bulb, it will spread to the other parts in a Parkinson's model.
So this is like a massive natural experiment, as you say.
And
I think you conjectured that maybe we would see increased rates of dementia going forward, right?
So where are we with that? Because we're 5 years in, right? And I want to be clear, I don't believe anyone is doing a test on humans worldwide. I think nature has provided the test, and we can treat it as a test and use it, to understand disease process or not.
But at the population level, have rates of reported, generative.
So here's the thing. The thing that made me finally write this paper after worrying for so long. At first, I was not worried because the olfactory receptors don't have the right receptor for the virus to get into them, but it can get in other ways. It can get in through the ensheathing cells and through the cerebrospinal fluid, through a capillary network. There are a lot of different routes. And if damage comes in, the olfactory bulb actually has an extra robust immune system to respond to insult, because this is a weak link in, stuff getting into your brain. So at the time, the thing that made me finally write the paper was the UK Biobank study. So the UK Biobank is this long-running program where they're collecting health data from, I think, a couple 100,000 people. And the subset of those, over 2,000, I think, are also periodically getting brain imaging. So they're looking at how things change over the lifespan. I think they are not testing smell, which is sad. So they had some subjects when they came back for their test, I think it was in 2021, for their imaging tests, had COVID in the interim. So they had about 400 subjects. So they age matched 400 other subjects, and they compared their brains. And most of these people had mild COVID, and they saw early signs of degenerative changes in areas connected to the olfactory system. Now, they didn't look at the olfactory system because that's not the way they do the imaging. To look at olfactory areas, you actually have to scan at different angles and do everything differently. So people normally ignore the olfactory system. So that's where we were left in fall of 2022, that there were mild degenerative changes about 5 months post-COVID. And I've had a student working on, a new analysis of the literature, looking through the papers that have been published since. And it looks like we're not done with the analysis yet, but it does look like this pattern is continuing, that in people who have some persistent COVID symptoms, the level of cognitive change correlates with the level of smell dysfunction. Well, people don't always know that they have smell dysfunction. Like I said, it has to get pretty bad before people think they can't smell. Like some people recovered their sense of smell and, assume that everything's normal, but they didn't recover it entirely. So there's some evidence that this pattern is continuing. Whether it's getting worse, I don't know yet, but certainly the amount of smell dysfunction correlates with the amount of cognitive dysfunction. And so it's like nature is sort of handing us an opportunity, kind of a silver lining, I guess. I don't really like to say that about tragedies, but like here's a chance to detect and figure out how to treat early cognitive decline. And the question is, are we going to take advantage of it, right? Now, I don't know for sure whether it's progressed or it looks exactly the same as before, and we don't know at all what the vaccination status is of people who had COVID who are having these problems, right? If they had COVID before they got vaccinated, we don't have those data. But I suspect there may be some interaction there, that the people who are not vaccinated are probably having a worse outcome here. But I don't have any data for that.
Interesting. I sort of asked ChatGPT the question really quick while you were talking. So, and it actually
Okay.
does refer to the UK Biobank data that survivors aged 50 plus had a higher likelihood of new onset vascular dementia compared to uninfected controls. But it does say that there isn't enough sort of evidence overall, but it is suggestive. So very, very interesting to see what happens in the years ahead, because you're right. I mean, this was a sort of a natural experiment, where nature threw the curveball at us and we were able to observe its impacts.
Yeah, the theory that this happens, through an insult through the nose, that neurodegeneration happens by that route, used to be kind of a joke. When about 30 years ago when people started seeing that this connection between smell dysfunction and neurological disorder. I heard somebody say, oh, what? So you get a cold and, 20 years later you get dementia? And no, because the olfactory bulb knows how to take care of that. Otherwise, we'd all be dead. But sometimes it can't, or sometimes its response is too big. So this is a perfect opportunity to figure that out.
Yeah, I guess we'll know in a few years. Yeah, but very fascinating. So before we wrap up, Leslie, one last question. We've been talking about consciousness. Are you aware of the link between smell and mood or happiness?
Oh, yeah.
What's that telling us? What's the research in that space all about?
Yeah, I think that the best way to talk about this, and you sent me a question, have I read Perfume? Yes, I read that book 20 years ago, and it was so intricate and terrifying that I just couldn't see the movie because I didn't want to ruin it.
It's not as good as the book, by the way.
Oh, okay. Well, I figured, but it's interesting. I have to see it to see how they managed to convey smell, right?
They say a picture is worth 1,000 words, but a smell, I don't know.
Yeah,
I don't know how you describe it in words. Which, by the way, is a whole other area
we could
of
totally
conversation.
Talk about that. Yeah.
That I've had with Alex Wilczko and some other smell people, right? Because you can describe smell without getting into biology at all.
You should talk to Asifa Majid if you want to talk about smell language. She's amazing, and she has studied it in people in languages where they have rich smell language. And it's fascinating, the difference in culture and the cultures in which you're supposed to smell other people's body odors, things like that. So happiness, one way to think about happiness is to think about unhappiness, right? As a scientist, we take things apart. And so the olfactory bulb is an integral part of the limbic system, right? And one of the best models for testing antidepressants is to remove the olfactory bulbs it's been done mostly in rats and mice, but it was done once in monkeys too, same effect. So it's not because they can't smell. If you cut the olfactory nerve, they can't smell, just like with taking out the olfactory bulbs, but they don't show signs of depression. So the animals, we don't know if they're happy or sad, but they show behaviors that look like things that humans with major depression do. They have behaviors that are, assigned as anxiety behaviors. They're afraid of brightly lit spaces more than they normally are. And they don't like sugar as much as they normally do. And that's the standard test for anhedonia, which is unhappiness, right? And so if you treat them with antidepressants, they get better at about the same speed as animals do. It's one of the only models that allows you to predict how fast an antidepressant will work. So we don't know exactly why this works, but I think thinking about the mitral cells in the olfactory bulb, they fire at about 10 to 20 times per second. That's about 10 times faster than most neurons in cortex. So with every inhalation, the limbic system is getting hit with this barrage of activity, right? Even when you're not smelling anything. And they're always going. And if you take away that background excitation, things are not going to work right. So Walter Freeman, I always tell my students, any question you have about olfaction has probably been answered 50 years ago by Walter Freeman with the equivalent of, technological equivalent of a coat hanger and duct tape. But he did this experiment where he cut the connection of the olfactory bulb to the olfactory cortex or piriform cortex and stimulated the remaining input to the piriform without the olfactory bulb.
Now, this was what, in rabbits?
In rats. This one was in rats.
Mm-hmm.
And the piriform cortex did not respond as it should. Normally, if you hit a shock stimulus to the olfactory tract bundle, it makes like an oscillation in the piriform cortex. If he took out the olfactory bulb, it didn't. If he replaced that activity with some background stimulation, like you might just have going on randomly, and then did a shock stimulus, the oscillation came back. So what that means, and then another graduate student did the same thing for the entorhinal cortex, which is part of the hippocampus. So when you take out the olfactory bulbs, these areas are out of their dynamic range. They're out of the range where they're supposed to function. Now, why that makes them sad, we don't know. It could just as easily make them happy. So happiness, right? What about happiness? I mean, we know, and this is where people ask me about aromatherapy, right? And it's a really slippery slope here because it can get very easily into kind of made up knowledge, anecdotal knowledge. But there are some studies showing that exposure to certain types of odors actually makes people happier.
So when you say people are spending more on scents than on tequila, that's money well spent, maybe?
Probably. I mean, I've become really, as I've started to become more interested in the stimulus that we use in our experiments, I've become very interested in perfumes. So I collect perfume samples, and I got to say, they make me happy. If I'm feeling sad, I just pick one up and smell it. But there are a certain class of chemicals that have been proposed to actually be especially calmness and happiness, I guess, is terpenes and terpenoids. And these are like the smell of pine trees, right? And so forest bathing, it's also present in plants in the mint family, lavender, rosemary, etc. And there are a couple of studies that were done with lavender and rosemary, and I forget which one where they had the subjects eat the substance. They swallowed it in pills. So dissociating the good smell from the effect on the body, right? And it does show up in your blood when you smell these terpenes. And the lavender, in one study, showed it made people more calm. You know how they give you the little lavender pillow for sleeping? Maybe that's a thing. And rosemary made people's memory better. But it also made them sleep better. And we know that better sleep gives you better memory. So it's possible that it was through that effect. But Greg Bratman at the University of Washington is studying the effect of terpenes from being in the forest on people's well-being. And it's really, it's early days, but it's really fascinating work. I certainly love to walk in the forest, but that's not science.
No, it's a great space. And, I've had 2 conversations with Alex Wilczko of Osmo, and now I'm beginning to have a conjecture as to why he always looks so happy,
Yeah.
and upbeat. He's surrounded by amazing smells. In fact, when I talked to him, it was face to face, and he actually had me smell a couple of vials that they had produced. Yeah,
I've become that annoying person who says, "Here, smell this." Even when it's bad, when I smell something, I annoy my wife by telling her, "Smell this, it's terrible."
Well, Leslie, it's been fascinating. Thank you for the time. I really enjoyed this conversation. I'm very, very interested in smell and its role in our lives, which I think is highly underappreciated. So I'm looking to, make some, good progress in this space. So thanks
Great.
So much.
And thank you for bringing attention to the sense. I think it's a good public service.
Cool. Thanks so much, Leslie.